All right, so the other procedure that we're going to be doing today is the biochemical tests, the first bit of it. For that, hopefully you've read the manual, you understand we're looking at different substrates and different products for them and what we're using to do it. This is the EnteroPluri Test. We just call them Enterotubes around here, or Enterotubes. And as you can see, 12 different chambers with different substrates in them. Come on, focus. Work with me here, phone. There we go. So you have glucose, lysine, ornithine, indole, adonitol. You can see them there. You can read them. Right, so each of these is a different substrate that will tell us things about the bacteria that we are investigating. So depending on whether or not it can metabolize these, we'll see color changes or other things happen, and then we can get a code from that. We'll talk about how to do the codes next week when we're interpreting these tubes, and we can use that code to figure out what species or the most likely species that we have. All right, to do that, we're using our EMB plates. You made some EMB plates of your pre and post. You're going to be sampling some of those and seeing what species you have, or the most likely one. In this case, this is one of the demos that I made, and you can see that there's a nice bit of stuff here. All right, so in theory, all of this should be the same species for what we're working with, but we're just going to pretend that it isn't, and we're just going to try and sample just one of them. Okay, so to do that, we have our tube. The tube's ends will unscrew. So you'll want to do that. You'll want to loosen each, but not yet remove them. Okay, so there are two ends to this. The blue end has the handle, and the white end does not. All right, the white end is the sampling end. And what you do is you take a look at your plate and you find an individual isolated colony, one that looks big and juicy enough that you can actually do something with it. But the way that stuff grows on EMB, it never really gets big and juicy. So just find one that is as big as you can get and that will work, well, relatively well. Okay, so go ahead and pick this one here. And you just sort of swirl it around, try and pick up as much of that stuff as possible. And you know what? We're going to go ahead and do two. Normally we wouldn't, but we want to make sure that we get results. And we know that these are all the same species. Okay, so now that you've got that, cover your plate back up, hold on to it. Okay, so what we do here is we pull this through. And what's going to happen is that it is now inoculating each of these chambers. So you just take it through each of them and you sort of twist this thing so that hopefully a few cells are getting into each of the chambers. You want to make sure that you get all the way, the tip gets all the way here to the glucose chamber. So maybe swirl it around a bit more at the end and then draw. Okay, once you've done that, you push it back in, and you want to leave the rod in here. The rod is sealing off between the chambers so that they can be anaerobic if they need to be. On the rod, you will see, and it's sort of hard to see on the camera, but there's a little notch right here, and that little notch, it is scored to be broken right there, and that's what you want to do. You want to bend it, and it will break. Okay. So at that point, or even before that, you put the lids back on. Once you've broken it off, you can put the blue one on. And this thing, don't touch the tip of it. It is still, hopefully, sterile or aseptic. You use it to poke holes in, and it's not really showing up on the camera. Come on. You can do it. I believe in you. All right. Well, you'll be able to see, but there are these little indents, and you poke holes in them with the end of this. You make sure that you don't touch the end because we aren't wanting to introduce any other bacteria, just the stuff that we have. The last 8 chambers have things that, there you go, have these little notches for you to poke the holes in. Once you've done that, you'll take some parafilm and you'll cover that up. Parafilm is a protective, it's wax basically, but it allows air to go through, but not anything bigger. And so it will allow, because we're poking these holes so that we have aerobic conditions in there, it'll allow air to get in without letting it become contaminated. Easiest way to end up messing up your results is to not have this be properly sealed. Other stuff gets in there, it starts metabolizing, and all of a sudden we're getting color changes that make no sense. Okay, so that's simple. Once you've done that, you put the parafilm on there. You'll write your name, your lab ID here, and then we're going to incubate these, and we will take a look at them later and see what the results are. Now, yeah, we'll talk about that in the lab. All right, and that is it for the biochemical test. Very simple, straightforward. It'll actually take you less time to do it than it took me to explain it.